Coming to next chapter, enzymes. So what are enzymes? Enzymes are biocatalysts, thermolabile, colloidal and protein in nature. So biocatalysts, they are catalyze the reactions in our body, and they are colloidal in nature, thermolabile. So they can be denatured by heat, thermolabile, and they are protein in nature. So most of the enzymes, they are protein in nature. They are nothing but proteins. So we discussed in proteins. So there are some proteins which have catalytic function, nothing but enzymes. So this is about the definition of enzymes. So enzymes, basically they have active site where the substrate binds. So when the substrate binds, so we call it as a binding site, and there it brings some chemical changes at the active site of the enzyme. So that is called as catalytic site. So active site has binding site, also called as catalytic site. So coming to classification of enzymes, according to International Union of Biochemistry and Molecular Biology, enzymes can be classified into 6 classes. They are oxidoreductases, transferases, hydrolases, ligases, isomerases and ligases. So this is how all the enzymes are classified into 6 classes. So based on the type of reaction they catalyze. Right. Coming to oxidoreductases first. So the name itself indicates the mechanism or the reactions catalyzed by this group of enzymes. So they catalyze oxidation reduction reactions. So enzymes catalyzing the oxidation reduction reactions are called as oxidoreductases. Right. So examples you can see lactate dehydrogenase, glutamate dehydrogenase, pyruvate dehydrogenase. So all the dehydrogenases are the examples for the oxidoreductases. So in oxidoreductases we have different types of enzymes like dehydrogenases, oxidases, oxygenases and reductases. So all these enzymes, they come under this classification, oxidoreductases. So basically they are catalyzing oxidation reduction reactions. So what is oxidation? Loss of hydrogens is called as oxidation. Otherwise gain of hydrogens is called as reduction. So loss of hydrogens is oxidation, gain of hydrogens is called as reduction. So addition of oxygen is called as oxidation, removal of oxygen is called as oxidation, right? So addition of oxygen is oxidation, removal of oxygen is reduction. Coming to transferases, so the name itself indicates here their function. Transferases, the enzymes catalyzing the transfer of groups. So whatever the group it may be, we call it as transferases, right? So for example, if you take aspartate transaminase, alanine transaminase, they are catalyzing the reactions of transferring of amino group. So there is transfer of group from one substrate to other substrates. So such type of enzymes which are catalyzing the transfer of groups are called as transferases. So hexokinase, the first reaction in case of glycolysis is catalyzed by glucokinase or hexokinase. Here it is transferring a phosphate group, right? So whatever group it may be, if you transferring a group, we call such type of enzyme as transferases. Coming to the third class, that is hydrolases. So enzymes catalyzing the reactions of breaking of bonds by introducing a water molecule, they are called as hydrolases. So hydro stands for water. So here the enzymes are breaking the bonds using water molecules. That's why they are called as hydrolases. So that bond may be a glycosidic bond, peptide bond, or ether bond, ester bond, whatever it may be. So whenever it breaks the bond by introducing a water molecule, we call those enzymes are hydrolases. For example, if you can take glucose 6-phosphatase, acetylcholinesterase, so those are the enzymes which require water or which introduce water and break the bonds. Coming to fourth class, ligases. Ligases are the enzymes which catalyze the reactions of breaking bonds without the addition of water molecule. They are called as ligases. Ligases also break the bonds but without adding water. So such type of enzymes we call them as ligases. So examples are aldolase. In glycolysis, So fructose 1,6-bisphosphate is converted to glycerol 3-phosphate and dihydroxyacetone phosphate. The 6-carbon compound, that is fructose 1,6-bisphosphate, is converted to two 3-carbon compounds, glycerol 3-phosphate and dihydroxyacetone phosphate. So here the bond is broken without the introduction of water molecule. So that's why you call it as a ligase. Right? So that's about the ligases, fourth class of enzymes. Coming to fifth class enzymes, isomerases. So isomerases are the enzymes which catalyze the isomerization of substrates, or the enzymes which catalyze the interconversion of isomers. So we call them as isomerases. Right? So examples are epimerase and racemase. So when you take epimers, glucose and galactose are epimers with regard to the fourth carbon. So glucose can be converted into galactose, galactose can be converted into glucose by the action of enzyme called as epimerase, because these two are epimers, a type of isomers. So epimers are nothing but a type of isomers, and these two are interconverted by the action of enzyme called as epimerase, and such type of enzymes which are catalyzing the reactions of isomerization of substrate are called as isomerases. So this is a fifth class. Now coming to the last one, ligases. So what are ligases? So ligases are enzymes which are catalyzing the reactions of linking together of two substrates, so which requires the supply of energy, and that is supplied by ATP. Right? So that is the basic thing what happens in case of reactions catalyzed by the ligases. They link two substrate, and for that linking of two substrate it requires energy supplied by ATP. So basically there is construction of bonds when the reaction is catalyzed by ligases. So there is a destruction of bonds whenever a reaction is catalyzed by ligases. So that is the difference between ligases and ligases. So ligases, they break the bonds, ligases, they create the bonds. Right? So for creating the bonds, for joining the two substrates, they require the energy supplied by ATP. For example, if you can take acetyl-CoA carboxylase. So acetyl-CoA carboxylase, it joins the acetyl-CoA and carbon dioxide. So for joining these two substrates, it requires the energy supplied by ATP. Right? So these are the 6 classes of enzymes, starting from oxidoreductases, we have transferases, hydrolases, ligases, isomerases, and ligases. Right? So this is about the classification of enzymes into 6 classes. Now coming to properties of enzymes. So as we discussed in definition, so enzymes are nothing but protein in nature. Right? So these enzymes, they have active site. Right? So there is small site cleft on the enzyme where the substrate binds. So that site is called as the active site. Right? So substrate binds to the enzyme at a particular site. So that site is active site. And you can see in this diagram picture, so the enzyme has a small cleft and substrate binds to that cleft and forms a complex called as enzyme substrate complex, and that gives rise to product, and enzyme is released in that reaction, and that enzyme reacts with to that enzyme one more substrate binds, and again products are formed. Right? So at the end of reaction, product is formed with the release of enzyme. So where this substrate binds to the enzyme at the active site. Coming to specificity. So each enzyme is specific in its reaction. So each enzyme binds to a particular type of substrate. So catalyzing only one type of chemical reaction, that is called as specificity of the enzyme. Coming to another property, cofactors. So what are cofactors? So we discussed that enzymes are protein in nature. So actually these enzymes, they have two parts, protein part plus non-protein part. So that protein part of an enzyme is called as apoenzyme, and the non-protein part of the enzyme is called as coenzyme or cofactor. So that cofactor, the non-protein part of enzyme, based on its association, we can classify that into coenzyme or cofactor. Right? So if it is a coenzyme means when that non-protein part binds with the enzyme in a loose bonds and loose association, then such type of uh, non-protein part we call them as coenzyme. If that non-protein part binds tightly with the enzyme, then we call that non-protein part as the prosthetic group. Right? So that prosthetic group is biotin. Biotin binds tightly with the enzyme carboxylase. So that's why you call it as biotin as a prosthetic group. And when you take coenzymes NAD plus pyridoxal phosphate, NADP plus FAD, all these coenzymes, they bind, all these non-protein parts, they bind loosely with the enzymes. So that's why we call them as coenzymes or co-substrates. Right? And if it is a metal ion, we call it as, if whenever a metal ion binds with the enzyme, we call it as metalloenzymes. Right? So like this, based on the type of association to the enzyme, we have classified non-protein part into coenzyme and cofactor. So when you see, so when an enzyme has both protein part and non-protein part, we call that enzyme as holoenzyme. So protein part is a coenzyme, non-protein part is called as coenzyme, and together we call it as holoenzyme. Coming to factors affecting enzyme activity. So the rate of reaction, velocity of a reaction is affected by various factors. So some important factors are concentration of enzyme, concentration of substrate, effect of temperature and effect of pH. So all these have effect on the velocity of the reaction or rate of the reaction. So concentration of enzyme. So whenever you provide the optimum temperature and pH, and when you increase the concentration of enzyme, the rate of the reaction increases proportionately. So here the rate of the reaction is directly proportional to the concentration of enzyme. So you increase the concentration of enzyme, the rate of the reaction also increases. Right? So this is a first order reaction. Coming to effect of concentration of substrate on the rate of the reaction or velocity of the reaction. So whenever you are increasing the substrate concentration, the rate of the reaction also increases up to a certain point. So initially the rate of the reaction is directly proportional to the concentration of substrate. Right? So that obeys the first order kinetics, and after reaching a particular concentration of substrate, so beyond that concentration there is no further increase in the velocity of the reaction, and from there onwards the rate of the reaction is, it obeys the zero order kinetics. How much substrate concentration you may increase, but there is no further increase in the velocity of the reaction, because at a particular concentration of substrate all the enzymes are occupied by the substrates. So when all the enzymes are occupied by the substrate, there is no further increase in the velocity of the reaction. So that point where all the enzymes are occupied by the substrates is called as the saturation point, because all the enzymes are saturated with the substrate. So at that particular point, whatever the velocity obtained is called as the maximum velocity. So we call it as velocity maximum. And so when you consider half of that maximum velocity, we call it as the Km value. So Km value is nothing but substrate concentration at half of the maximum velocity is called as the Km value. Coming to significance of Km value, so Km value has some significance. That is, some enzymes have high Km value and some enzymes have low Km value. So what do you mean by that? High Km value and low Km value. What happens to an enzyme with high Km value and low Km value? So when you consider, but for example, you consider hexokinase, it has a low Km value towards its substrate glucose. Means even at low concentration of substrate, it can bind with the enzyme and it can be converted into glucose 6-phosphate. Right? So it can be half saturated even at the low concentration of the substrate. It can attain half of the maximum velocity at low concentration. So when you take glucokinase, it has high Km value for glucose. That means only at high concentrations of the substrate, that is glucose, it can convert glucose into glucose 6-phosphate. So that is the significance of Km value. So significance is whenever an enzyme has high Km value, it has low affinity towards the substrate. Whenever an enzyme has low Km value, it has high affinity towards substrate. Right? So that is the significance of Km value. Now coming to effect of temperature. So what happens at various degree of temperatures. For example, most of the enzymes in our body, they are active in the range of 35 to 40 degrees centigrade. Means they show maximum activity within this temperature. So at extremes of temperature, so when it is beyond that 40 degrees centigrade, 45 degrees centigrade, then what happens? The activity of enzyme decreases. So that is the effect of temperature on enzyme activity. So at a particular temperature, the enzyme shows maximum activity. So that temperature where it shows maximum activity, we call it as optimum temperatures. And at extreme temperatures, enzymes are subjected to denaturation, and whenever denaturation of protein occurs, it loses its biological activity, and whenever it loses its biological activity or loses its structure, it leads to decrease in the velocity of the reaction. So that is the effect of temperature on rate of the reaction. Coming to effect of pH. So the hydrogen ion concentration also affects the rate of the reaction or velocity of the reaction. So whenever pH is changed, it affects the ionic charge on the proteins. So that's how it may increase or decrease the velocity of the reaction. Right. So when you take, each enzyme has a specific pH at which it is active, and the pH at which an enzyme shows maximum activity is called as optimum pH. And at extremes of pH, the activity of enzyme, the rate of the reaction decreases because of denaturation of protein. So when you take, for example, pepsin, so pepsin is active in the range of 1 to 2 pH. So in the acidic range, pepsin shows maximum activity, that is when the pH is 1 to 2, it shows maximum activity. And in the basic range, like salivary amylase and pancreatic amylase, they are active in the basic range. So 7 to 8 salivary amylase, and at the range of 10 pancreatic amylase is more active. So each enzyme has a particular pH at which where it shows the maximum activity. So that pH we call it as optimum pH, and optimum temperature is the temperature at which it shows the maximum velocity. So and extremes of temperature and pH, the activity of enzyme decreases. So because of the denaturation of enzymes. So those are the factors affecting the enzyme activity. Effect of concentration of enzyme, concentration of substrate, temperature and pH. Now coming to mechanism of enzyme action, how these enzymes will act. So that mechanism of enzyme action can be viewed in two different perspectives, that is energy changes occurring during the reaction and the chemical changes that occurring at the active site of the enzymes. So we have to consider these two. So by that, so we can examine the mechanism of enzyme actions. Coming to energy changes which occur at the during the reaction, that is, so actually the substrates have energy and products have different energy. So we have to convert the substrates from low energy state to a high energy state. So from ground state we have to shift the substrate to a high energy state. That shifting of substrate from the ground states to a high energy state called as transition state requires the energy. So that energy difference between the substrate state and this transition state is called as the activation energy. And the function of enzymes here it is, they decrease this energy, that is activation energy. So whenever that activation energy is decreased, the rate of the reaction is rapid. So the rate of the reaction increases. So whenever the activation energy increases, then the rate of the reaction decreases. So The function of enzymes is they decrease the rate of the reaction so that substrate molecules are shifted to the transition state, and whereby from this transition state they are converted into products. So that is the energy changes which occur during a reaction. So enzymes, they decrease the activation energy, the energy required to shift the substrate molecules from the substrate state to the transition state. And coming to chemical changes which occur at the active site, so they are general acid-base catalysis, covalent catalysis, transition state stabilization, and the other one is proximity effect. So all these four are the chemical changes that occur at the active site of enzyme. So coming to general acid-base catalysis, so when a substrate binds to the active site of enzyme, there's the residues at the active site of the enzyme, they can donate the protons or accept the protons. So that type of chemical change we call it as acid-base catalysis. Coming to transition state stabilization. So whenever a substrate binds to the active site of enzyme, so the active site of enzyme favors the conversion of substrate and transforms that into a transition state. So whenever a substrate is transformed into a transition state, it is favoring the conversion of substrate into the product. So that is called as transition state stabilization. Coming to covalent catalysis, where the active site, the amino acid residues at the active site, they may donate or they may add some groups to the substrate, and whereby they favor the catalysis. So that type of mechanism we call it as covalent catalysis. Coming to proximity effect, so this proximity effect is nothing but here the active site of the enzyme provides some space for the interaction for the substrates. So whenever it creates a space for the interaction between the substrates, so whenever that interaction is increased, it's nothing but the rate of reaction is increased. So it is favoring the condition, or it is favoring the condition by increasing the interaction between the substrates so that they can be transformed into products, they can be converted into products. So these are the four chemical changes that occur at the active site of the enzyme. So that is the conversion of substrates into products. Right. Coming to enzyme substrate complex formation. So enzymes, they convert substrates into products. Right. So here, whenever a substrate binds with the active site of the enzyme, it forms a complex called as ES complex. It is nothing but enzyme substrate complex. So once ES complex is formed, so most of the times it is converted into product and with the release of enzyme. Right? So there are some theories which explain this ES complex formation, enzyme substrate complex formation. Those are lock and key model or Fisher's template theory. Here, this lock and key model explains that each substrate has a prefixed enzyme active site, so that it states that basically the lock and key theory states that the active site of the enzyme is a rigid structure. Means, so each active site of enzyme has a specific substrate, so that only binds to the enzyme. So it states that the active site of the enzyme is rigid. It is a prefixed one, but actually in actual the active site of the enzyme is a flexible one. So that's how that is not accepted nowadays, and the more accepted theory is induced fit theory. So induced fit theory explains that so whenever a substrate approaches the active site of the enzyme, it brings some conformational changes in the active site of the enzyme such that the substrate binds with the enzyme at the active site, or whenever the substrate binds with the active site of the enzyme, it brings some conformational changes in the active site of the enzyme so that it favors the binding of more substrate. So this is called as induced fit theory. So here substrate is inducing changes at the active site of the enzyme to bind with the enzyme. Right? So that's why it is called as induced fit theory. So according to this theory, the active site of the enzyme is a flexible part. It's not a rigid one. It is flexible in nature. So that's how whenever a substrate with a different conformation approaches the enzyme active site, the conformation of the active site is changed. So this is the more accepted one, induced fit theory. Right. So those are the two theories which explain the mechanism of enzyme substrate complex formation. Right. Now coming to enzyme specificity. So enzyme specificity is because of the active site of the enzyme, and here there are three types of specificity. Those are stereospecificity and reaction specificity and substrate specificity. So three types of specificity are there: stereospecificity, reaction and substrate specificity. What is stereospecificity? So we discussed that we have two types of amino acids, D amino acids and L amino acids, and enzymes, they are specific to each type of amino acids, each type of proteins. So there are some enzymes which act on only D amino acids, and there are some enzymes which act only on L amino acids. For example, amino acid oxidase, D amino acid oxidases, they act only on D amino acids, and L amino acid oxidases, they act only on L amino acids. So this is stereospecificity. Coming to reaction specificity, a particular substrate can be converted into different products when it is acted upon by different enzymes. For example, when you take transaminases, they convert, when they act on a particular amino acids, they transfer the amino group, and the same amino acid undergoes decarboxylation, same amino acid undergoes transmethylation. So same amino acid is undergoing different reactions but catalyzed by different enzymes. So such type of specificity is called as reaction specificity. So same substrate is undergoing different reactions catalyzed by the different reactions. Coming to substrate specificity. So there are some enzymes, like for example if you take urease, so that urease has only one substrate, urea. So urease cannot act on glucose, urease cannot act on any other amino acids, urease cannot act on fatty acids, like so urease has its a particular substrate, that is urea. So urease acts only on urea, not on any other substrate. So that is called as substrate specificity. So these are the three types of specificity shown by the enzymes. Now coming to inhibition of enzyme activity. So generally substrate binds with the enzyme, it forms the enzyme substrate complex, gives rise to product. But in some reactions there are some compounds which inhibits the enzyme activity or which decrease the rate of the reaction. So such type of compounds we call them as inhibitors. So what is an inhibitor? Any substance which decreases or halts the enzyme activity, we call such type of substances as inhibitors. And we have two types of inhibitions: reversible inhibition and irreversible inhibition. So whenever the effect of the inhibition can be reversed, inhibitor can be removed by dialysis, so dilutions. So such type of inhibitions are called as reversible inhibitions. If the inhibitor cannot be removed, so such type of inhibitions we call them as irreversible inhibitions. Right? So that is the basic difference between reversible and irreversible inhibition. That is, so whenever you can remove the enzyme, you can remove the effect of inhibitor by dilution, we call them as reversible, and if you cannot remove that effect of inhibitor, we call them as irreversible inhibitions. Coming to types of inhibition, we have different types of inhibitions like reversible inhibition, irreversible inhibition and allosteric inhibition. And in reversible inhibition, again we have different types of inhibition. So those are competitive inhibition, non-competitive inhibition and uncompetitive inhibition. So basically in reversible inhibition, the inhibitor binds to the enzyme by non-covalent bonds. In case of irreversible inhibition, the inhibitor binds to the enzyme by covalent bonds. So there is strong bonding between inhibitor and enzyme in irreversible inhibition, where there is loose association between inhibitor and the enzyme in the reversible inhibition. Okay. So this reversible inhibition is three types: competitive inhibition, non-competitive, and uncompetitive. Coming to competitive inhibition, so what is competitive inhibition? So the name itself indicates there is competition. So competitive inhibition is basically there is competition between substrate and inhibitor for the active site of enzyme. So this competition arises because of similarity in the structure between substrate and inhibitor. So because of that similarity in the structure between substrate and inhibitor, they compete for the active site of the enzyme. So that's why we call it as competitive inhibition. So there is competition, and that competition is because of similarity in the structure between the substrate and inhibitor. So because of that similarity in the structure, the inhibitor in case of competitive inhibition is called as substrate analog. So that competitive inhibitor is called as substrate analog. So you can see, so whenever the concentration of substrate is more, that substrate binds with the enzyme and forms the enzyme substrate complex and gives rise to product. But whenever the concentration of inhibitor is more than the substrate, and there in that condition the inhibitor binds with the active site of the enzyme replacing the substrate and forms the enzyme inhibitor complex, and that enzyme inhibitor complex cannot give rise to products. So this is what basically happens in case of competitive inhibition. So whatever the concentration is more, if the concentration of substrate is more, it forms the ES complex and forms the product. If the concentration of inhibitor is more, that inhibitor competes for the active site of the enzyme, because as the concentration of inhibitor is more amount of inhibitor binds to the active site of the enzyme, it forms a complex that cannot give rise to product. So most of the drugs, they act by this mechanism, competitive inhibition. Right? So in this picture you can see this competitive, whenever the concentration of inhibitor is more, it is binding with the enzyme and forming the EA complex. Right? And what happens to the Km value and velocity maximum in case of competitive inhibition? Right? Km value, we discussed that Km value is the substrate concentration at which it gives half of the maximum velocity, is called as the Km value of the substrate. So in case of competitive inhibition, there is no change in velocity maximum. So velocity maximum is unchanged, but there is increase in the Km value. So here the reason is, in the presence of inhibitor, we have to increase the concentration of substrate so that it attains the maximum velocity. So whenever you are increasing the concentration of substrate, that means the Km value is also increased. So that's why in case of competitive inhibition, so there is increase in Km value with unchanged velocity maximum. So that is the effect of a competitive inhibitor on velocity maximum and Km value in case of competitive inhibition. Right? So in this picture you can see, so there is no change in velocity maximum, but there is change in the Km value, that is increase in the Km value in the presence of competitive inhibitor. Coming to some important examples, you can say examples of citric acid cycle enzyme succinate dehydrogenase. So that succinate dehydrogenase converts succinate into fumarate, and that succinate dehydrogenase is inhibited by another molecule called as malonic acid. So that malonic acid has similarity in structure to that of substrate. So here malonic acid is a competitive inhibitor and succinate is the substrate, and the enzyme is succinate dehydrogenase. So whenever you increase the concentration of malonic acid binds with the succinate dehydrogenase and it cannot form the fumarate. So there it is inhibiting the reaction. And whenever you increase the concentration of succinic acid, it forms the enzyme substrate complex and forms the fumarate. So that is the best example you can give in case of competitive inhibition, malonic acid inhibiting the succinate dehydrogenase. So that's about the competitive inhibition. Now coming to another type of reversible inhibition, non-competitive inhibition. So there is no competition between substrate and the inhibitor, but still there is inhibition. So that's why we call it as non-competitive inhibition. So inhibition is there, but there is no competition between substrate and inhibitor. Yeah, so in competitive inhibition, we have seen that the substrate and the inhibitor, they compete for the same site, active site of the enzyme. But in case of non-competitive inhibition, what happens is, here the inhibitor won't affect the binding of substrate to the active site of the enzyme. So inhibitor binds to the enzyme other than the active site, and it won't affect the binding of the substrate. So in case of non-competitive inhibition, both inhibitor and the substrate, they bind to the enzyme at a different sites, right? So that is what basically happens in case of non-competitive inhibition. But whenever the inhibitor binds with the enzyme, it forms a complex called as enzyme substrate inhibitor complex, right? So that inhibitor may bind with the enzyme after the formation of ES complex, otherwise before binding of substrate. So whenever inhibitor binds, that forms EA complex, and to that substrate binds, ESI complex is formed. So that ESI complex cannot give rise to products, right? So this is what basically happens in case of non-competitive inhibition. So inhibitor won't affect the binding of substrate to the enzyme. Still there is inhibition because of formation of ESI complex formation. So ESI complex cannot give rise to products. So what happens to the Km value and velocity maximum in case of non-competitive inhibition? In case of non-competitive inhibition, so as we have seen, in case of competitive inhibition, there is increase in Km value because we are increasing the concentration of substrate to attain the maximum velocity to overcome the effect of inhibitor. But in case of non-competitive inhibition, and inhibitor is not affecting the substrate binding to the active site of the enzyme, it is not interfering with the substrate binding. So there is no change in Km, means it is not affecting the affinity of the enzyme towards the substrate, but there is decrease in the velocity maximum because of the formation of ESI complex, there is non-functional enzymes. So there is increased non-functional enzymes because of ESI complex, so that leads to decrease in the velocity maximum. Right? So that is what happening in case of non-competitive inhibition. So there is no change in velocity Km value, but there is decrease in the velocity maximum. Coming to examples of non-competitive inhibition, we can see example in glycolysis, fluoride inhibits the enzyme enolase. So that is example for we can take under non-competitive inhibition. And other examples are, in case of some examples are heavy metal ions like mercury, lead. They inhibit some enzymes by non-competitive inhibition. So they react with the sulfhydryl groups of present at the active sites of some enzymes and inhibit the enzymes by non-competitive inhibition. Right? So this is about non-competitive inhibition. Coming to the third type of reversible inhibition, uncompetitive inhibition. So what happens in competitive inhibition? So here the inhibitor binds to the enzyme only after the formation of ES complex. So after formation of enzyme substrate complex, then only inhibitor binds with the enzyme. Right? So that is the mechanism of uncompetitive inhibition. So in the picture you can see, first substrate binds with the enzyme and forms the ES complex. So then only the inhibitor binds and forms the ESI complex that cannot give rise to products. Right? So this is the basic mechanism that occurs in the uncompetitive inhibition. And so what happens to the Km value and velocity maximum in case of uncompetitive inhibition? Here both Km value and Vmax are decreased in case of uncompetitive inhibition because unfunctional enzymes are decreased, non-functional enzymes are increased. So whenever non-functional enzymes are increased because of formation of ESI complex, the velocity maximum decreases. And why this Km value is decreased? Km value is decreased because of, so here the specificity, the significance of the uncompetitive inhibitor is it favors the more binding of substrate to the enzyme, so that then only the uncompetitive inhibitor can bind with the ES complex. So it favors more binding of substrate to the enzyme. So that's why there is decrease in the Km value and that results in the formation of ESA complex, so the velocity maximum also decreases. The both Km and Vmax are decreased in case of uncompetitive inhibition. And the example is the alkaline placental alkaline phosphatase is inhibited by the enzyme, is inhibited by a compound called as phenylalanine, and that type of inhibition is uncompetitive inhibition. So you can see, so in this picture, so there is decrease in both Km value and velocity maximum in case of uncompetitive inhibition. Right. So till now we have seen reversible inhibitions, reversible competitive inhibition, non-competitive inhibition and uncompetitive inhibition. So whenever the inhibitor binds covalently with the enzyme, then it leads to a irreversible inhibition. There the inhibitor binds tightly with the enzyme, so we cannot dissociate that inhibitor from the enzyme by dilution or whatever so mechanism it may be. So such type of inhibitions we call them as irreversible inhibitions. So there is one compound called as nerve gas, that is diisopropyl fluorophosphate, so that inhibits the enzyme acetylcholinesterase by this mechanism called as irreversible inhibition. Right. So that's about the inhibition of enzymes. Coming to regulation of enzyme activity. So there are different ways in which the activity of enzymes is regulated, and those are allosteric regulation, feedback regulation, and regulation by activation of latent enzymes, covalent modification, induction depression, and the other mechanism is compartmentation of metabolic pathways. So what is allosteric regulation? Allosteric regulation is basically we have active site where the substrate binds, but in addition to that active site we have an additional site on the enzyme that is called as allosteric site. So that allosteric site, where there is a molecule called as allosteric modulator binds. So to that allosteric site of enzyme, the additional site of enzyme, allosteric modulator binds, and based on the type of modulator, the enzyme activity may be increased or decreased. If the allosteric modulator is a positive modifier, means whenever the allosteric modulator binds to the allosteric site, it increases the activity of enzyme by bringing some conformational changes at the active site of the enzyme, so that it allows the more binding of substrate to the active site of enzyme. Okay? So that is called as a positive modifier. So positive modifier increases the activity of enzyme. And the same way, when a modulator, negative modifier binds to the allosteric site, it decreases the activity of enzyme by bringing the conformational changes at the active site of enzyme such that it prevents the binding of substrate to the active site of enzyme. Right? So in these two ways, the activity of enzyme may be regulated, may be increased or decreased by a allosteric modulator. So this type of regulation is called as allosteric regulation. For example, if you take carbamyl phosphate synthase 1, CPS1 in case of urea cycle, so CPS1 activity is increased by N-acetyl glutamate. It is a positive modifier, means whenever N-acetyl glutamate binds with the carbamyl phosphate synthase 1, the activity of enzyme is increased. So more amount of carbon dioxide and ammonia will be converted into carbamyl phosphate. So that is a positive modifier. So whenever negative modifier, for example, if you take citrate synthase, so citrate synthase activity is decreased by ATP. So whenever the concentration of ATP is more in cells, so that ATP binds with the active site of the enzyme and it decreases the activity of enzyme, that is, it decreases the activity of citrate synthase. So this type of modifier is called as a negative allosteric modulator or modifier. So that's about the allosteric regulation. Coming to feedback regulation, what happens in feedback regulation? Generally in feedback regulation, the end product of a pathway inhibits its first enzyme. So that type of regulation is called as the feedback regulation. For example, you can take heme synthesis. In heme synthesis, ALA synthesis is the key enzyme, the first enzyme in the heme synthesis, and that is inhibited by the heme, the end product of heme synthesis. So that is feedback regulation. Same way, cholesterol synthesis, cholesterol inhibits the HMG-CoA reductase, and that mechanism of regulation is nothing but feedback regulation. Coming to another type of regulation, activation of latent enzymes. So some of the enzymes in our blood, they are secreted in, they are present in inactive forms, right? So whenever there is requirement, it leads to proteolysis of that enzymes is brought and they are activated. For example, chymotrypsin and trypsin, right? So those are the enzymes activated by this latent activation of latent enzymes is, for example, is chymotrypsin and trypsin. Coming to covalent modification, what is covalent modification? And covalent modification basically involves addition or removal of a group may increase the activity or decrease the activity of enzyme. So such type of modification is called as covalent modification or covalent modulation. So simply you are adding a group or removing a group. So whenever you add a group or you remove a group, it may increase or decrease the activity of enzymes. So that is covalent modification, and the most common type of covalent modification is phosphorylation and dephosphorylation. So you are adding a phosphate group or removing a phosphate group from the enzyme. So such type of reactions are called as phosphorylation and dephosphorylation. Glycogen synthase enzyme in the glycogenesis is inactivated by phosphorylation. So when you add a phosphate group to glycogen synthase, the activity of enzyme is decreased. And when you take glycogen phosphorylase, the enzyme, key enzyme of glycogenolysis, the activity of enzyme is increased whenever you add a phosphate group. So it is called as phosphorylation. So when phosphorylation of glycogen phosphorylase is increasing the activity, phosphorylation of glycogen synthase is decreasing the activity, right? So this type of increasing or decreasing the activity by adding a group or removing group is called as covalent modification. Now coming to induction and repression, another type of regulation. So this occurs at the gene level. Okay? For example, you take insulin. So whenever insulin is secreted, whenever there is increased glucose levels in the blood, that insulin is secreted. That insulin increases the rate of synthesis of enzymes of glycolysis. So insulin increases rate of glycolysis by increasing the concentration of the key enzymes of glycolysis. How the key enzymes of glycolysis are increased, concentration is increased, that is by called as induction. So induction is nothing but you are increasing the concentration of enzymes by increasing their synthesis. So that synthesis of enzymes is increased and that is brought at gene level. So insulin increases the activity, de-represses the enzymes of the glycolysis, key enzymes like glucokinase, phosphofructokinase and pyruvate kinase. So these three enzymes activity concentration is increased by inducing the genes which code for these three enzymes. And again repression, this also occurs at the gene level. Here the opposite mechanism occurs. There you are decreasing the activity of enzymes which code for the enzymes, right? So when you are increasing the activity of enzymes which code for the enzymes, we call it as induction, and so opposite mechanism we call it as repression, right? So this is another type of regulation of enzyme activity. Coming to compartmentation of metabolic pathways, so this is another type of regulation of enzyme activity. Here in compartmentation of enzymes, it's nothing but, so some of the pathways are located in cytosol and some of the pathways they are operated in mitochondria. For example, if you take fatty acid oxidation that occurs in the mitochondria, whereas fatty acid synthesis occurs in the cytosol. So these two pathways, the enzymes concerned with the synthesis of fatty acids are present in cytosol and oxidation are present in the mitochondria. So this is how these two pathways can be regulated, and that type of regulation is called as compartmentation. So these are the ways in which the activity of enzyme is regulated. Now coming to isoenzymes. What are isoenzymes? So we know coenzymes. Coenzymes are the non-protein part of the enzymes, we call them as coenzymes. Then what are isoenzymes? Isoenzymes are the multiple forms of an enzyme which catalyze the same reaction, we call them as isoenzyme, right? Multiple forms of the same enzyme. So same enzyme exists in different forms, but all those forms of enzymes they catalyze the same reaction. So such type of enzymes we call them as isoenzymes or isozymes. So So these isoenzymes or isozymes, they have different structure, they show different electrophoretic mobility, they have different optimum pH, they have different Km and Vmax values, and susceptibility to the inhibitor is different for the isoenzymes, and the degree of denaturation is different for the isoenzymes. So these are the ways in which the isoenzymes, they have different properties. So they differ in the structure, electrophoretic mobility, degree of denaturation, susceptibility to inhibitors. So those are the important points regarding the isoenzymes. Coming to examples for the isoenzymes, the examples are lactate dehydrogenase, LDH. So what is lactate dehydrogenase? So this lactate dehydrogenase interconverts, catalyzes the interconversion of lactate and pyruvate. So it can convert lactate to pyruvate, pyruvate to lactate. So here, so this lactate dehydrogenase is nothing but a isoenzyme. So why you are calling it as isoenzyme? Because it exists in different forms. Like it has 5 forms: LDH1, LDH2, LDH3, LDH4 and LDH5. So all these forms of LDH, number 1 to LDH5, they can catalyze the conversion of pyruvate into lactate, but they have different properties. So what are those properties? What is the difference between LDH1, LDH2, LDH3 and LDH4, LDH5? So what makes LDH1? What is the difference between LDH2? What are the basic differences between all these isoenzymes of LDH? So LDH1 has 4 subunits, 4 polypeptide chains. So each polypeptide chain is called a subunit, and all the subunits are of H type. So we are naming that subunit as polypeptide as H, right? So H stands for heart, right? So H subunit. When all the subunits of same type, that is, all the subunits are H, and if all the subunits are H type, we call it as H4. It's nothing but LDH1, right? So that LDH1 is mainly present in heart, right? And when you subject to electrophoresis, the fastest moving fraction in electrophoresis is LDH1, and its concentration is 25%. And coming to LDH2, its composition is H3M, means there are 3 subunits of H type and 1 subunit is of M type, and it is present in RBC. And coming to electrophoretic mobility, it is faster, right? Fastest is LDH1, faster is LDH2, and its concentration is 35%. So when you see concentration of LDH1 and LDH2, the concentration of LDH2 is more than LDH1, right? So there is a clinical significance for this concentration of LDH1 and LDH2 that we will discuss later. Coming to LDH3, so its subunit composition is H2M2, and it is present in the brain and kidney. And coming to, it is the fast fraction in the electrophoresis. And coming to its mobility, it has, its concentration is 27%. LDH4, its composition is H1 and M3. It is the slow fraction in electrophoresis. And coming to LDH5, it has 4 subunits of M type, means all the subunits are of similar type, that is M type, M4. And it is the slowest fraction in the electrophoresis, and it is present in the skeletal muscle, right? And it accounts for the 5% of total LDH activity. So that's about the isoenzymes of LDH. There are 5 isoenzymes from LDH1 to LDH5. So these isoenzymes are because of different composition, different subunits and different composition, right? So coming to, so as we discussed in the properties of LDH, they differ in their Km value and velocity maximum. When you see LDH5, it has low Km value for its substrate, that is pyruvate, means even in the low concentration of pyruvate, it can convert pyruvate into lactate. And the LDH1, which is present in heart, it has high Km value towards its substrate pyruvate. So that is significance of LDH1, LDH5 Km value. And coming to diagnostic importance of LDH, so it is one of the cardiac markers, cardiac enzymes. The activity of LDH is increased. The level of LDH is increased in case of myocardial infarction. Right. So as you discussed, the concentration of LDH1 is more. LDH1 is 25% to 30%, and whereas concentration of LDH2 is 35%. So concentration of LDH2 is more than LDH1. But in case of myocardial infarctions, this concentration is reversed. That is, concentration of LDH1 will be more than LDH2. So this pattern is called as flip pattern. Right? So there is reversal of concentration of LDH1 and LDH2. So we call it as flip pattern. Right. So concentration of LDH1 starts to rise within 12 to 24 hours, and it reaches to a peak value by 3 to 4 days, and it remains in blood up to 10 days. So that is the time period for the LDH lactate dehydrogenase in the blood in case of myocardial infarction. Coming to another enzyme which has isoenzymes, that is creatine kinase. So this creatine kinase, also called as creatine phosphokinase, is another isoenzyme, another enzyme with isoenzymes forms. So this creatine kinase converts phosphocreatine into creatine, creatine into phosphocreatine. Right. So CPK, the creatine phosphokinase, has two polypeptide chains, two subunits. So it is a dimer. Dimer means it has two monomeric units, two subunits, two polypeptide chains. So those polypeptide chains are of two types. It may be of B type or M type. B means brain and M means muscle. Right. So CPK1 has BB subunit, and it is present in brain. So B stands for brain, it is present in brain. CPK2 or CKMB, it is present in heart. So its subunit composition is MB, it is present mainly in heart. Coming to CPK3, CK3, its composition is MM, and it is present in the skeletal muscle. So this is about the creatine kinase isoenzyme forms, and the first enzyme to elevate in case of myocardial infarction is the creatine kinase. It starts to rise within 3 to 6 hours and reaches a peak value by 24 hours and remains in blood for 3 to 4 days. So that is the clinical significance of creatine kinase. So and the CKMB, it is mainly estimated in case of myocardial infarction because its concentration is increased in case of myocardial infarction. So that's why it is called as cardiac marker. So this is a more reliable marker than LDH. Right. Coming to, so here you can see in this picture the onset of the time period in which the levels of LDH, creatine kinase and AST, these three are called as the cardiac markers. Their level is elevated in case of myocardial infarction. Each marker has a different time interval in which it rises. If the graph under the curve is more, it is proportional to the myocardial infarction, right? So another isoenzyme is alkaline phosphatase. So this alkaline phosphatase removes the phosphate groups, that's why it is named phosphatase. So there are 6 isoenzymes of alkaline phosphatase: alpha 1 ALP, alpha 2 heat labile, alpha 2 heat stable, pre-beta and gamma ALP. These are the important isoenzymes of alkaline phosphatase. So what is the significance of alkaline phosphatase? So that alpha 2 heat level alkaline phosphatase suggests the hepatitis. So in case of hepatitis, the concentration of alpha 2 isoenzyme is elevated, and same in case of bone diseases, pre-beta ALP is elevated in case of bone disease. So that is the clinical significance of alkaline phosphatase. Thank you.